So we left off last class. We talked a lot about the discovery of DNA as the genetic material, and we talked about the structure of DNA, how it's a double helix with bases in the center. We talked a little bit about base pairing rules. We're going to start today talking about how DNA functions to code for proteins. So you know that DNA is genetic material in that it provides a recipe book for how to make proteins. And how it occurs is through two processes. So a gene is a sequence of DNA that encodes for specific proteins. So we'll often be talking about the gene for this, the gene for that. And what we mean by that is this is the gene, this is a region of DNA that has the recipe for how to make a protein that's involved in something or other. So you have a gene for making each of the enzymes that we've talked about in previous chapters somewhere in your DNA. So the process of using genes to make proteins occurs in the first step is called transcription, and the second step is called translation. And we're going to talk mainly about transcription today, and then we'll get into translation probably on Friday. Transcription is the process of taking DNA and converting it into a similar but slightly different molecule called RNA. Translation is the process of taking RNA and converting it into protein. So the reason why this happens in a two-step process like this is in part to protect the DNA. So translation, the process of making proteins, is going to happen out in the cytoplasm, so either bound to the endoplasmic reticulum or freely floating in the cytoplasm. And out here, there are a lot of enzymes called DNases that will destroy DNA. So if DNA was to get out of the nucleus, it would start being degraded by enzymes out there. So we keep it protected inside our nucleus. So part of the reason why eukaryotes have a nucleus is to keep that material basically on reserve. It's like reserve material in the library. It should be a little more protected than books that are allowed to circulate, the same idea. But we need to get the recipes out here, so the first thing that happens is we copy the DNA sequence into RNA. We're also going to learn that part of the reason why you make RNA is to control the production of proteins. So RNA is something that we can actually do a lot to control how long we make the proteins, where we make the proteins, when we make the protein, and things like that, which are going to allow for a lot of control of the functions of our cell later on. So after we talk about transcription and translation, we're going to move into talking about regulation of genes and regulation of protein production. And some of that involves controlling the transcription step or controlling translation step, and even controlling how long RNA lasts once it gets out into the cytoplasm. So there's lots of reasons why this happens in two steps, and we'll get into some of those as we go along. Okay, so again, the two steps of making a protein are transcription and translation. Transcription is when the cell is going to copy the gene's DNA sequence into an RNA sequence. So it's basically like you took your cookbook and you wrote down the recipe. And now you're going to take that recipe to your kitchen and make the brownies in this example. The process of taking that recipe and actually making a protein is translation. So the information in RNA is used to manufacture a protein out of amino acids. So first you copy the recipe, and then you read the recipe and follow it to make the protein. All right. So before we get into some of the details of transcription, we're going to talk a little bit about what RNA is and how it is different than DNA. Okay, so The main difference that we covered several chapters ago between RNA and DNA is the sugar. So RNA has a sugar called ribose as part of it, and DNA has a sugar called deoxyribose. And you'll see they're almost exactly the same. The only difference is that deoxyribose has a hydrogen here, whereas ribose has an OH here. So deoxy means without an oxygen. It's one less oxygen. So DNA has ribose, or RNA has ribose instead of deoxyribose. DNA has deoxyribose. Another difference is in the bases. So all three of them, all DNA and RNA have three of the same bases. They all have adenine, they all have guanine, and they all have cytosine. So A, G, and C. The last base here, thymine, is only in DNA. In RNA, there's a very closely related, similar base called uracil. So it's similar in structure, slightly different. But other than that, the other three bases are the same. And also, DNA is a double-stranded molecule, whereas RNA is generally single-stranded. It's a single-stranded molecule. It's one strip. Okay, so where this is going to be important is in the base pairing rules. So DNA, A pairs, when you're going from DNA to DNA, A pairs with T, A pairs with T, and G pairs with C, right? But if you're going to be making DNA into RNA, you have A, G, C, and T. Because there's no thymine in RNA, A is going to pair with U, G with C, C with G, and T with A. So you have just a slight change to things. So the base that's complementary to adenine is uracil in RNA. And the base that's complementary to G is C, C is G, T is A. It's all the same. Okay, so it's a little bit more complicated, and we're going to see that when we actually look at transcription occurring. Okay, so just a few little differences between them. Okay. RNA comes in three different flavors, and we're just kind of setting this up because we'll talk about these a lot more when we get to translation. There's three different kinds of RNA. There's messenger RNA, which we'll spend the bulk of our time talking about today. Messenger RNA, or mRNA, is a copy of the recipe. It is the form of RNA that carries the information or the recipe for making a protein. It contains groups of three bases called codons on it. So you can break up messenger RNA into certain codons. So that's really the main type we're going to talk about today, and that's what I've showed you in the last two slides ago, where I showed the DNA being copied into RNA and then RNA then being read to make a protein. That was messenger RNA we were looking at. But in order for a protein to be made, it's going to involve two other kinds of RNA as well. There's ribosomal RNA, or rRNA, which actually physically makes up the ribosome. So we've talked about a ribosome way back in, it was chapter 3, I think. Ribosomes are the site of protein synthesis. They're actually physically made out of ribosomal RNA combined with proteins. So that's actually an enzyme that helps synthesize RNA based on the messenger RNA recipe. And in addition to interacting with ribosomal RNA in terms of the ribosome, we're also going to see a third kind of RNA called transfer RNA, or tRNA, which will actually be the thing that physically reads the message in messenger RNA. tRNA, transfer RNA, will bind to the mRNA codons, the mRNA sequence, at one end of the tRNA, and at the other end it's going to have an amino acid with it. So by pairing up transfer RNA and mRNA, you're going to bring in the appropriate amino acid. And we'll go into this in a lot more detail when we get to the translation step. So tRNA, rRNA, and mRNA will all interact together to make a protein, ultimately. So they're all going to be very important. So I just wanted to set up that there are 3 different kinds, although today we're going to mainly talk about mRNA. Though the process that we're looking at today does occur for all 3 of them. They're all made in the same way. We really kind of look at this from the mRNA perspective because it's kind of the first step in protein synthesis. Okay. Okay, so you have a long double-stranded region of DNA, so double-stranded DNA, and you want to copy a certain portion of it. So let's say, grab these other markers. Okay, so here's DNA. It's double-stranded, double helix, like this. Nope, that was not very good. Okay. So you've got double-stranded DNA, and let's say this region right here is a gene, is a recipe for making a protein. You're going to first take this recipe and copy it into mRNA. And that's the first step that we're looking at right now, which is called transcription. Transcription is going to occur in 3 basic stages. There's initiation. You need to be able to access this DNA. Elongation, which is the process of taking the DNA and copying it into RNA. And then termination, you're going to have to stop making the RNA. So pretty straightforward. So we're going to look at these in a little more detail. So you're going to take a DNA template. You're going to initiate, or first unwind the DNA and open it up. Elongation is the process of making mRNA here. And then termination and release is ending the process at the end. Okay. So the first step, initiation. The very simplest explanation for this is just that an enzyme that is going to eventually make RNA, which is called RNA polymerase. So enzymes often end in ASE. The enzyme polymerase makes a polymer of RNA from a DNA strand. Initiation occurs at a region on the DNA called the promoter. So if this is the gene here that you want to copy the recipe from, just before that gene, just before the region that you want to copy, is a special region of DNA called a promoter. And when we get to gene regulation, the promoter is going to be very, very important. There's a lot of things that go on at the promoter region that we'll talk about later on when we get to gene regulation. The promoter is a site of most regulation for gene production. DNA polymerase doesn't just randomly lie down on any gene. It needs to be signaled as to where to start making RNA. And a lot of that happens at the promoter. We're going to talk about something called a transcription factor and other proteins that will sit down at the promoter region and signal to RNA polymerase to come over and bind here. But we won't get into that really today. So RNA polymerase will bind to the promoter region, and that initiates transcription. The promoter region is going to be, we're going to see as a more accessible region. The RNA polymerase is going to be able to unwind and separate the 2 strands of DNA at that location and start making RNA. Okay. So once RNA polymerase binds, it can then start running along one of these two strands and making RNA. So this would be the elongation stage. So after RNA polymerase binds to the promoter, it will start moving along the DNA and adding nucleotides to the 3' end of the growing RNA strand. So remember what I said last class, that RNA and DNA can only be made 5' to 3'. So 5' to 3', which means that this RNA strand here that's being made in this direction, this would be the 5' end of that RNA strand. This would be the 3' end, and it's being manufactured in that direction. So this template strand, this DNA, what end of the DNA strand is this? Is it the 3' or the 5'? 3. Yeah. So this would be the 3' because they're going to be opposite, and this will be the 5' of the DNA because they are going to be opposite of each other. So you're making the RNA 5' to 3', and it does this by using complementary base pairs. So this system here, anywhere there is a C in the DNA, you put a G in RNA. Anywhere there's a G in the DNA, there's going to be a C in the RNA. Anywhere there's an A, there's going to be a U. And if this was a T in the DNA, the next base put here would be an A, right? So one by one, RNA polymerase will add new nucleotides as it runs along the DNA template, making a RNA strand. Okay, yep, so this will keep going until it reaches another specialized region of DNA called the terminator sequence. The terminator. And the terminator sequence will signal the end of the gene. So the promoter sequence signaled the start of the gene. The terminator sequence here signals the end of the gene. When RNA polymerase runs into that terminator sequence, it will separate from the template and release the new RNA strand. So now you have a brand new RNA molecule that's been made here that is ready to move on to the next step. Okay, so again, fairly straightforward. We're overlooking a lot of complexity, some of which we'll get into near the end of the chapter when we talk about what's actually going on at that promoter sequence. But basically, promoter signals the start of the gene, RNA polymerase binds to the promoter, initiates transcription. RNA polymerase will run along the DNA strand, making the RNA in the 5' to 3' direction. So this would have been the 5' end of the RNA. This would be the 3' end. And it's following the complementary base pairing rules, A to U, G to C, C to G, T to A. When it reaches the terminator sequence, RNA polymerase will fall off. The RNA will separate, and you'll now have RNA. Okay? Simple enough? Straightforward. Okay. So, this isn't the only thing that's going on in the nucleus, however. Prior to the RNA leaving the nucleus and meeting up with a ribosome to be translated, something else will happen to it called RNA processing. So the RNA is going to be processed before it leaves the nucleus. And the major things that we're going to talk about in this class as to what happens to RNA, and there can be other things as well that go on to it, is that there is going to be strands of RNA added to both ends of the RNA. There's going to be a 5' cap put on the RNA strand, and there's going to be a 3' poly A tail, and something called an intron is going to be removed, or introns are going to be removed from the molecule. So the cap and the tail are mainly to protect the RNA. Introns have kind of a odd reasoning for them and slightly not really well understood reasoning for them. And we'll get into that in a minute. So 5' cap and the poly A tail. 5' cap is just an addition of a very short sequence of modified nucleotides to the 5' end of the RNA. The poly A tail is an addition of 100 to 200 adenines to the 3' end. Any guess as to why you add 100 to 200 adenines to the end of mRNA? What that might do for your RNA? All right, yeah, that's a good guess. It's not going to be complementary to anything. All right, it's way past the stop codons. It's this extra kind of long trailing bit at the very end of it. All right, yeah. So if you remember what I said, what happens to DNA if DNA was to leave the nucleus? Someone's either read ahead or it was a good guess. All right, excellent. Excellent, yeah. So what happens, as soon as DNA would leave the nucleus, it would start getting destroyed. Same thing happens to RNA. As soon as RNA leaves the nucleus, there are destructive enzymes that will start chewing away at it. And most of those enzymes, which are called RNases, will chew away at the 3' end. Turns out that the 5' end, a lot of things can't attack from that side. So that's why you only put the small little cap on it. The 3' end is more vulnerable, and it's going to start RNases are going to start chewing away at those adenines. So let's say you wanted a protein to be made for a very long time. Would you put 100 adenines or would you put 200 adenines on the end of it? All right, 200. It should make sense. The more adenines you add to the poly A tail, the longer this message is going to stay in the cytoplasm, and the longer it can be made into a protein. Let's say, on the other hand, you wanted to have very, very fine control as to when you would stop making a protein. Would you want a long tail or a short tail?
A short tail.
Why a short tail? Why would that? Okay, so you want it to stop at a very specific time. So if you have a very short tail, you know that once you stop making that mRNA, in a very short amount of time, the protein is going to stop being made. Right? So your cell can stop making RNA and then protein will stop. If you have a really long tail, there's a lag period between when you stop making the RNA and when you stop making the protein. So there's some proteins that your body, your cells, want to have very fine control as to when they start making the protein and when they stop making the protein. And so they're actually going to make pretty unprotected mRNA. They're just going to constantly produce that mRNA. Then as soon as they stop making the mRNA, this mRNA will be destroyed and it will no longer make a protein. And this is one of the kind of quirks of this system, is that as soon as this mRNA is sent out into the cytoplasm, it's going to be read and made into a protein. It's basically the machinery that makes proteins are pretty blind to what protein they're making. They're going to read any recipe and just start cooking it up. And so you need a self-destructing message so that eventually they stop making that protein, or else you would be making all the proteins that you've ever made all the time. Right? So this is another reason why to use mRNA. We basically have these throwaway messages that we can produce and then allow to be destroyed. So there's a lot of other stuff that is in RNA, and this graphic kind of shows that there are these what are called untranslated regions, UTRs, polyadenylation signals. This actually tells the enzyme that's called poly A polymerase, which is stuff I don't expect you to remember after this, but it's interesting. This tells the poly A polymerase how many A's to add, so it's a polyadenylation signal. There are sequences in the 5' UTR and the 3' UTR that tell different proteins where to take the mRNA. So some of this mRNA is going to head to ribosomes on the endoplasmic reticulum. some mRNAs will head to ribosomes in the cytoplasm. Some might actually go into the mitochondria, and there are ribosomes in the mitochondria that might make the protein from there. So there's a lot going on in what are called these untranslated regions. What we're going to see kind of next is, after I talk about the other processing thing, is what's going to happen here, which is in your protein coding segment. So actually, when you make mRNA, and this is something that will be important when we get into gene regulation, you don't just copy the recipe. You also copy stuff that's before the recipe, and you copy a bunch of information past the recipe, all of which we used to think were just junk DNA. They were untranslated DNA. But we now know that they serve a lot of purposes. So they determine how long that protein is going to be made. They determine where that protein is going to be made, and they determine a whole bunch of other stuff as well that we won't really get into. Okay, so 5' cap, 3' poly A tail. Primary purpose is to protect the mRNA, to make it last for a certain amount of time out in the cytoplasm. Okay. Another kind of odd thing that happens to mRNA before it gets sent out to be translated is the removal of introns. So introns are portions of the mRNA that are going to be removed prior to translation. The remaining portions, the portions that stay behind that are left of the message, are called exons. And within that protein coding sequence within the RNA, there could be these large stretches or large regions that don't actually code for the protein that need to be removed. So an enzyme called a spliceosome will recognize certain markers on the RNA, cause the RNA to fold, it will cut out the intron region, and it'll stitch together the exon. So these two parts will be stitched together, and this lighter pink part will be cut out. Why this happens is still a little bit of a mystery. So this would be like if you wrote a 50-page essay, and then right before you handed it in, you took out 30 pages from the middle, threw them out, put together the rest of it, stapled it together, and handed it in. So you're doing a whole bunch of work for nothing, basically. And this is the same thing. Your RNA polymerase made all of this RNA that's just going to be thrown away. So why that happens, don't really know. We're going to talk a little bit later on in this chapter about what's called differential splicing. And that is one thing that this helps with, is that you can actually take the same mRNA and make multiple recipes from it by cutting out different regions of it, and we'll talk about that. That could be one thing. But the majority of these, that doesn't even occur to. It's the same exact piece that's cut out every single time. So how this came about, how this evolved, is a mystery. But it might explain how mutations often arise in different proteins and different enzymes, which is important, and we'll talk about that later on as well. So intron removal, another way that RNA is processed. Okay, so here's a question to see how much you've followed so far. So let's say you have a DNA template strand with the following sequence, A, G, T, C, T, T. What would be the nucleotide sequence of the complementary RNA molecule produced by transcription? And don't worry about 5' and 3' here. I haven't put that in, but we will talk about that in a minute. Okay? You guys very quick? Seems very easy. That's good. So most of you got it right. Pretty much all of you put E or well, 81% of you put E. Why is C wrong? So some of you put C, but what's wrong with C? Okay, so if I had said, what is the complementary DNA strand to this DNA strand, then C would have been the right answer. But I asked for the RNA, and RNA does not have thymines in it. So the T here needs to be a U. So that's the difference between C and E. Some of you also chose B, and what you did is you just turned the T's into U's. But remember, complementary means the one that it would be based on, which means that if there's an A, the RNA that is complementary to it is going to be a U. Complementary to a G would be a C, complementary to a C would be a G, complementary to a T would be an A. So you don't just take the T's and change them into U's, you actually follow the base pairing rules. So A would be U, G, C, T would be A, C would G, and the two T's would be two A's. So good. Okay. So good. So if I was to put directionality on this, if this was being made in this direction, what side of the RNA would this have been? So if this RNA was being made in that direction, which direction is RNA made? 5' to 3'. So this would have to have been the 5' end, and this would have been the 3' end. So from the DNA template, what side of the DNA template would this be? This would be the 3', and this would be the 5'. Right? So thing that trips up a lot of students is, oh, I'm making it 5 to 3, so I have to read this that way. But no, be careful. You're making the new strand 5 to 3, so it follows the template 3 to 5, basically. So the template strand you would be building in this direction. And this will become more important when we start looking at DNA to DNA, which we'll get into in the next chapter. But okay. Okay, so that's transcription. We now have mRNA that we've processed. I'm going to go a few slides into translation, but we'll talk more about that next class. But we have lots of time, so we'll go a little bit into this. So we've gone from DNA to RNA. That's transcription. That all occurs in the nucleus. That RNA was processed, so introns were removed, 5' cap, 3' poly A tail were added to it. The next step is translation. So translation is going to take the information that is in that mRNA and use it to build proteins from amino acids. So the process of translation, of building a protein from mRNA, is a little more complex than transcription. We're not just doing complementary base sequences. We're actually going to use a code system. So we're going to approach this today and Friday from two perspectives. One, we're going to do this like a word problem, like you're decoding something, and we're going to have a big table, which I'll show you, which we may or may not get into today, that lists the different codons and the different amino acids that correspond to them. And so we'll talk about it from that perspective. But we're also going to learn about this from the cell's perspective. So how does the cell actually read the code biologically? Because you don't have some little person with a clipboard and a coding sheet saying, nope, put a lysine next, put a serine next. There's a biological way that this works as well inside the ribosome. So the genetic code is a set of rules that a cell uses to translate mRNA into amino acids, and we're going to see that that really relates directly to those tRNA molecules that we said. tRNAs are the code breakers of your cells. A codon is a group of 3 mRNA bases that codes either for an amino acid or for a stop signal or something called a release factor. So turns out that the genetic code is written in words that are 3 letters long. So one of the reasons why scientists didn't at first believe that DNA could be the genetic material is that we only have 4 bases. We have A, G, C, and T. And they thought, well, it's ridiculous. With A, G, C, and T, how many words can you make? Well, it depends on how big your words are. If you only had one-letter words, which a lot of them thought originally, you could only make 4 words. And we know we have 20 amino acids. If you had 2-letter words, you could make 16 words from 4 letters. Still not enough. So it turns out 3-letter words is really the minimum we could have to make those 20 amino acids. And that's how it functions. So 3 letters, like A, A, G in RNA, are going to code for an amino acid, in this case, lysine. And the process of translation will be reading those 3-letter codes to make a protein. So through a series of many, many different kinds of experiments where researchers actually took very boring RNA, like poly-U RNA, which is U, 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 and then tried to make a protein out of that. They were able to determine the genetic code, and we actually have a nice little sheet. This is the one in your book, and you're going to use this today. You're also going to use this on the test. I'll give you copies of the genetic code like this. So every 3 letters codes for a different amino acid. How you use this sheet, the first letter of the codon is on the left here, U, C, A, or G. The second letter is up on the top, U, C, A, or G, and then the third letter is on that side. So if you had U, 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 you would look for the U, which is going to be everything that starts with a U is in the top 4 rows. Everything that is U, U is in this top left box here, because that's U here. And then U, U, U would be up here on the top, and it's phenylalanine. It's actually a little hard to read probably for you, but phenylalanine, which we're going to use the 3-letter code mostly for this, so PHE. And I'll give you a sheet that's a little bit clearer than this on the test, but it's the same idea. So if I had the codon CCU, what amino acid would that code for? All right, so C is here, C is here, CCU is here, so it's proline here as well. All right. So how about AGU? All right. So serine, yeah, it's a little hard to read from the back, right? Serine. So you're right. I need to get a better sheet. Yeah. Yeah. That's what happens with this classroom. Okay, serine. How about UAA? Stop. Yeah. So what do you think happens when that's encountered? Okay, stops making a protein. Right. Let's see. There's the other ones here. How about AUG? Yeah. All right, this says start and methionine, which is hard to read. So what do you think happens at AUG? Okay, so we're going to see that there is a signal in the mRNA as to where to start making a protein. And what do you think that signal is? All right, the code AUG. And there's a signal as to where to stop making a protein. And what do you think that signal is? All right, UAA, UAG, or UGA. So there's 3 versions of the stop sign. What amino acid do you think every single protein that your cells make initially starts with? Yeah, methionine here. So this doesn't just code for start, it codes for an amino acid called methionine. So actually every single protein that your cells make starts with methionine. There's things that happen after the proteins are made, and sometimes the first few amino acids are cut off. But when it was initially made, every single protein started with methionine. Okay. So let's just, we'll go into this in more detail next class, but let's see if you can read this well enough, or have a neighbor that has a book that has it in better sense to answer this question. So, no. All right. Okay. AUG, UCU, GUU, UGG. Can you turn that into a 4 protein? I'll get you guys a better one that shows up better on the board next time. All right, 100% of you said. Okay, well we can do this. So yeah, definitely I will fix this. Yeah, well there you go. There's a good start. Is that AUG codes for methionine, and you'll know that if you don't know that already. So AUG is here. It's methionine, so it's got to be one of these two. The next 3, UCU, would be up here, and it's serine. So you know it's not this one, so it has to be that. And then GUU would be here, and that's valine. It's very blurry, but I can see it. And then UGG is up there. That's tryptophan, and that'd be TRP. So you're right. So B is the best one. So I need to come up with a better one. Okay, so I'm going to stop here because I need to redo some of these slides and come up with more readable graphics. But what we'll talk about on Friday is how biologically this does, and we'll do a few more of these kind of problems to get you doing it. All right?